co-op was working. Okay. My very first co-op job placement as a first-year undergrad in systems design engineering was with Stuart Foster, doing some software programming for this very rudimentary high-frequency ultrasound machine back before they had even spun it off into the company and when they were still based out of Sunnybrook. And then most of my future co-ops at Waterloo were with Chris McGowan downtown at Sick Kids, who most of you probably know, doing some MR research. And that led to me knowing about MVP and being pretty intimately involved, and then decided to pursue my PhD with Graham Wright. Graham, unfortunately, let me work very closely with some interventional cardiologists as we were doing a lot of scar tissue mapping of the heart. And I got to know Sandy Dick and John Graham and Kim Connolly, and ended up being seduced by the idea of being a physician and doing clinically motivated research from that end. And so then I went to med school and stayed in Toronto, was based out of Sunnybrook for all of med school. So again, my first co-op was in the year 2000 here at Sunnybrook, and then started in 2004 with Graham, was full-time at Sunnybrook, went to med school and was based out of Sunnybrook, did my residency in radiation oncology, split between PMH and Sunnybrook. So I've been around for a long time, based mostly at Sunnybrook. And when I was in med school thinking about what I was going to do and still use all the skills and from my engineering and my PhD, radiation oncology is actually what was quite clearly the best fit. Academic career, they do a lot of imaging and physics research, and so that's what led me to do that. And there are no primary malignancies of the heart, and so the CNS is very MR heavy. Brain and spine relies on MRI, and so that's where my career led. So I'm going to present a lot of other people's work. We work as a team very well here at Sunnybrook, and the 5 other CNS or central nervous system radiation oncologists, led by Dr. Sehgal, who is sort of internationally renowned brain and spine oncologist, sort of leads the vision, and then the rest of us help guide the clinical research. And we work very closely with a number of people on MVP, Greg Stanish, and then all of his minions or people who have graduated from Greg's lab and are their own PIs now. So I'll present a lot of slides directly from Angus, Rachel, and Colleen, and Wilfred and Greg, and are heavily involved in the brain and the prostate research as well. And then we work with a big team here as well with radiation physicists, therapists, and other researchers. I am on call this week, which means it's been a little bit of a rough week. If I say stuff or rush through stuff that doesn't seem to make sense, please, you can raise your hand or just jump in and cut me off with a question. I'm more than happy to pause and answer any questions that come up as we go. There will be time at the end, but if it makes sense to ask something as I'm describing something, please use the raise hand function or just speak up. So again, this is the sort of radiation oncology 101. So about 3/4 of my time is spent in clinic talking to patients and describing, what kind of cancer they have, what are the options for treatment, why do we recommend radiation or not recommend radiation, what's the goals, what are the side effects of radiation, what's the follow-up. That's about 3/4 of my clinical time. And then the other, quarter or 25% of my clinical time is the back end of radiation oncology, which we call contouring, or when I describe it to my wife, it's drawing circles or coloring. So when we decide that we're going to treat a patient, so this is a prostate case, a patient either can't or doesn't want to have surgery for their prostate cancer, and then radiation is an excellent option that in many cases has the exact same cure rate. And so if a patient, let's say with an intermediate prostate cancer, decides they want radiotherapy, nowadays stereotactic radiation, which just means a high dose per day in a few number of days, is very commonly done. for the prostate. So we use 40 gray or 40 units of radiation in 5 fractions. So 8 units of radiation a day, every other day for 2 weeks. And in order to deliver these high doses safely, we use both a CT. So the LINAC machines that we use to radiate patients is CT based. So right now we need a CT because the actual attenuation of the photon beams is based on Hounsfield units from a CT scanner. So everyone right now gets a CT scan, and then in some areas of the body, like the prostate and the brain, we also commonly do an MRI, just because the contrast and ability to delineate the area we want to treat is so much better with MRI. So this is a patient who has an intramucous prostate cancer, wants stereotactic radiation. We bring him in, we get it's called a planning CT on the left and an MRI on the same day, but you can see that the patient actually passed a little bit of urine, therefore the bladder is a little bit smaller on the MRI. We fuse these together in order to contour or draw our targets. In this case, our target is the prostate, and you can see it very well on the MRI. We also have to delineate all the organs around that might have consequences from the radiation, or we want to make sure they don't get overdosed with radiation. So the prostate is mostly the bladder here and the rectum behind. Prostate sits right in between them. So we have to delineate slice by slice our volumes. We then pass off the volumes to our radiation therapists or the dosimetrists, and they're the ones responsible for turning these volumes and the prescription. So I say I want 40 gray of radiation here, and we need to limit the bladder to, X percentage of the bladder can't get this much radiation, and X percentage of the rectum can't get this much radiation. Otherwise, we know that the toxicities go up. And the therapists, in prostate, there's something called VMAT, where this red describes the arc of the beam. So there's a radiation, the head of the LINAC produces photons. They travel through an opening, and these are MLCs, or multi-leaf collimators, that can move in and out. And you can see it can create sort of complex shapes with allowing the radiation to pass through. And for prostate, the head of the LINAC slowly rotates 360 degrees around. And as it's doing that, it can speed up and slow down. And it can also, the MLCs can change and create all these complex shapes. And at the end of the day, we want to deliver the radiation dose to the target and minimize dose to the organs around it. And it's called inverse planning. So these therapists define all these sort of rules. And then the computer tries to optimize a plan by determining what shapes are delivered from all these various angles in order to get the final dose distribution that we want. So back in, before we had these sort of complex systems, you could do what's called a 4-field box, where a beam would come in anteriorly, posteriorly, from the left and the right, and where they overlap would give high dose, but there'd still be medium dose splashing out. IMRT is where you use beams from a bunch of different angles. In this case, it's only 3, but usually it's 9 or 11. And again, it gives you a better ability to control where the dose goes. And VMAT is an arc where, like I just described, and again, it has different properties in terms of being able to deliver dose to the area you want to treat. And so the dosimetrists will send us back a plan with what's called isodoses, where this is again, the blue shaded is the target of the prostate plus a margin. And we want to get 40 gray to sort of the middle of it, which is this red line. And we want 36.5 gray to cover well the outer edge of the blue target. And you'll see there's low dose splash coming out, and we evaluate these lines and how much dose is hitting the bladder and the rectum. And if it looks good, we approve it. Patient comes in on the day of radiation. They lie on this table here, this black table here, which can raise up and goes under the head of the LINAC. Now, we're doing very conformal high dose radiation. And even though we have the laser system and tattoos on the patient to make sure that they're where we think they are, internally, the bladder may not be the same size, the rectum may not be the same size. And so this is called a cone beam CT on the sides. Coming out here in red is a cone beam CT. And so the patient goes under. We do a cone beam CT, which is a very quick low dose CT that doesn't give great detail, but usually shows us good enough what we need to see so that we make sure that where we think we're treating is actually where we're treating and that the prostate is in the right location. And then if they're good to go, the radiation plan is delivered, again, for prostate every other day for 5 days. Now, much of the talk is going to be about CNS. And very similarly, when we do CNS, MRI is really the backbone. You cannot see brain tumors very well on CT. This is a glioblastoma, and lots of what I talk about will be glioblastoma, which is a grade 4 incurable brain tumor that arises from within the brain. It's what Gord Downey had, unfortunately, and passed away from. And the standard is either a biopsy or surgery to remove as much as possible, but unfortunately, it's never possible to remove all of a glioblastoma, even if visually you think you did. There's always tumor left behind and it always comes back. It is incurable. But what we do after surgery or biopsy is we do radiation, which helps and on average gives people about a 1.5 to 2 year lifespan. Without radiation, people don't make it to a year. But as you can see, this is a multifocal, so different areas of glioblastoma that you just can't see on a CT. But again, we bring patients in. We do a CT because the LINACs right now require a CT in order to calculate a dose distribution. We also do an MRI in order to determine all of our volumes. And this is what's called the GTV, or gross tumor volume, which is the gross tumor and or surgical cavity. Oh, okay, sorry. And so the red is called a gross tumor volume. We then know the standard way to treat GBM, well, there's a few different standards, is to expand around this by 15 millimeters, because we know there's always microscopic disease that extends out beyond the visible tumor. We also know this is a FLAIR image. I think with this group, I don't need to explain T1 and T2 and FLAIR. So this is a FLAIR image, and you can see that there's some FLAIR hyperintensity, which in GBM, we never know if it's just edema from the irritation from the tumor nearby, or whether actually there are microscopic tumor cells causing this FLAIR signal. But we usually include most of the FLAIR, especially if we're convinced that it's microscopic tumor, plus a minimum 15 millimeters. And you can see in this case, it ends up being a very large volume that we have to treat in order to deliver either 60 gray in 30 fractions, or in some patients, we use 40 gray of radiation in 15 fractions with an oral chemotherapy pill called temozolomide. And again, without the benefit of this MRI, we would have no idea where we need to treat. So one quick thing in terms of MR research, this is really research being driven by the companies that we work with. In this case, it's Philips, where something called MR-CAT, which means instead of actually doing a CT and using the Hounsfield units, which the LINAC machines require, you can do an MR acquisition and use the data from an MRI to create a pseudo or synthetic CT. So this is a CT that's derived completely from an MRI pulse sequence. And therefore, if we're starting to use this, actually the first case that we did it without doing a CT at all in the prostate was just about a week or two ago. And the hope is that if we can do this, we can get rid of the CT part, which, we have 4 CT scanners in our radiation department that are running all day, every day, doing these planning CTs in order to help map out radiation. So that's something that MRI can be used to get rid of the CT scan and eliminate one step. So the MR LINAC, so that's sort of radiation oncology 101 and how most of the world does radiation oncology. You do a mapping CT and a mapping MRI, and you define your volumes and you come up with a plan. And usually a week or two after that, you bring the patient in and you deliver the same plan every day for either 5 days or 30 days or however many days you need to treat. The group in the Netherlands at Utrecht started working on this technology 20 or 30 years ago, where they wanted to combine an MRI machine for its superior contrast with a radiation delivery machine like a LINAC. But the engineering behind it is very, very complex. And it wasn't until a few years ago that in partnership with Elekta that they actually produced and are now selling this as a clinical tool. So this is the machine. It is an MRI, 1.5T MRI with a LINAC essentially built around it. And this is our MR LINAC that's here over at Odette. So it was 2013 that they started to really think that this could be a viable clinical enterprise. We were one of the 10 founding members of this consortium that worked with the company to get these built and installed and up and running. And when I started on staff in July 2019 is when we got certification, and we started treating patients just about 2 years ago. And again, it's a really a technological marvel with a lot of complex interactions between the radiation beam and the magnetic field, where the radiation is affected by the magnetic field that's always on with MRI, as you guys all know. And so the modeling of it and taking that into account when we're delivering our radiation is something that took a long time to sort out. And so now we have a machine where we can do MRI simultaneously with delivering radiation. And so we've done a lot of brain cases here. We still bring them in and we do a pre-plan. We get a baseline or a planning CT and an MRI. We draw our volumes. We come up with a plan to deliver, 60 unit gray of radiation to this the glioblastoma here. And then the patients come on day one of their treatment. And instead of just blindly giving the same plan we did, we do a new MRI. Again, to start off with, we do a non-contrast MRI, a T1 and a flare. And we adjust in case that there's any shifts or rotations. We can compensate by adjusting and creating a new radiation plan based on what their anatomy and position is on that day, and deliver that plan instead of a reference plan. And we can do this daily. And so that's something called adaptive position, which we've been doing now for 2 years. But there's obviously a lot more we can do than simply do an MRI and adjust for any shifts in the patient's anatomy on any given day. And that's what I'll talk about. So a lot of what motivated our desire to start treating gliomas or GBMs on the MR LINAC is the fact that we can see with an MRI the tumor and the flare and how it's evolving day by day. And this study, okay, so I'll get to the study that motivated a lot of our work after the next slide. But again, just to reiterate, patients have surgery or a biopsy. On week 0, they get 5 days of radiation a week over 6 weeks with this drug called temozolomide. Again, up front here, they get a single MRI and a single CT, and the same plan is delivered every day for 6 weeks, every weekday for 6 weeks. And then 4 weeks after that, we do the first post-treatment MRI, which is the new baseline. And then patients can continue on chemotherapy. And eventually when they recur, might have more surgery or more radiation or more chemo. But that's the standard of how this is treated worldwide. There's a typical volumes of a glioblastoma and the CTV or clinical target volume that we target to treat. But we know that 10% of patients with glioblastoma, by the time we do their MRI 1 month after, the disease is already grown and gotten out of hand, whereas other patients do much better. And nobody had really understood exactly what's happening to the tumor and to the edema around the tumor during radiation. You get one scan before, you treat, and then you get a scan after. So we started this program about 2 years ago. This research study led by Sten Meyerhog, and the analysis was all done by James Stewart, looking at day 10 and day 20 of a 30-day course of radiation and 1 month after radiation, what is happening to these tumors. And this is a case where in white is outlined the original tumor. Day 10, you can see there was tumor extending beyond where we originally contoured. By day 20, it was extending to the contralateral side of the brain. And 1 month post, the disease was rapidly progressing. And again, our treatment would have targeted this original white area. If we don't know what's going on, we can't see that there's tumor growing beyond, and we're not treating that. And that's why it's growing and continuing to grow, because the original volumes are what are used for the entire 6 weeks. So we now looked at I think almost 100 patients, but this paper was on the first 61 patients, where we looked at both the growth or the volume of the tumor as well as the migration distance. If the tumor stays the same size, but moves by a centimeter, we're still missing it. And so the y-axis here is the relative volume, and the x-axis here is migration distance. And all these cases in the top right here are cases where the tumor is shifting outside of where we originally were treating. We want everything down in this left corner where the tumor is shrinking or stable and not moving. And that's why some of these cases don't do well, because they're growing and we're not treating them properly. So that's where the MRL comes in. And this is a study called United that I'm actually leading, and working closely with all my colleagues here, where people have talked about adaptive radiotherapy, and there's textbooks on adaptive radiotherapy, which just means instead of doing one plan and treating a whole course with that one plan, you adapt on the fly. And historically, that meant maybe once in the middle of radiation, you do a new set of scans, and if things have changed, you then come up with a new plan, which is better than nothing, but is not truly adaptive. We know things are changing more than just once every 2 or 3 weeks. And the MRL allows us to do daily MR imaging and really see what's going on. We can't do contrast every day because people's kidneys would not be happy about that, but we can do non-contrast MRI every day. And what this trial is doing once a week GAD injection, so that we have a full contrast enhanced MRI. And the goal of what we're doing is to use smaller margins. So on the right here, this is the tumor in red, and in green is a 5 millimeter expansion, and in yellow is the standard 15 millimeters expansion. So most places in the world would use at least a 15 millimeter, if not a 2 centimeter or 3 centimeter expansion, leading to a big volume here. And the reason for that is that we know there's microscopic disease, but also we know that this tumor could evolve, and therefore you need a big safety margin so you're not missing tumor. If we're doing daily MRI and we know we can see the tumor at least once a week, and we can see sort of subtle changes without contrast every day, then our thinking is we can use much tighter volumes, and that spares a lot of toxicity. When we use these big volumes, patients have a rough time getting through radiation. There's a lot of side effects, and I'll show later that some of the toxicity of radiation may actually be a reason why patients don't live as long. So our hope with this study is we're using 5 millimeter margin. We're doing truly adaptive radiation. It's really the first time that this machine is being used for its intended purpose, and the first time a real daily adaptive radiation trial is being done, with the hopes that we're not going to have tumor growing outside of this 5 millimeters, because we're going to adjust our volumes every day and every week, and hoping to reduce toxicity. And so you can see that our volumes are 50% or more less by doing these smaller expansion. And this is the very first patient on the trial, a patient of mine. This tumor is actually shrinking during radiation. So the volume is 45 cc's on the reference plan, and by day 6, it was 38 cc's. And we can reduce our volumes. We don't need to treat outside if things are responding nicely. And this is just another example to show how the tumor in red and a 5 millimeter expansion is in yellow. Now, we don't just limit it to 5 millimeters because there is some edema here that the oncologist wanted to include because they thought that there was microscopic tumor here. And so you can extend your CTV, we call it, but these volumes are still smaller than a 15 millimeter expansion. And so it's still a 20% difference in this case, even when you sort of extend beyond the 5 millimeters, which we know we sometimes have to do. But this illustrates the point that this flare signal, hyperintensity, we don't know if it's pure edema or if there's actually microscopic tumor in there. And I'll talk about how we're trying to get around that and figure that out in some future slides. And again, the motivation for this is a study from India that they compared two different methods for treating GBMs. What's called the RTOG method uses really, really big volumes. And the MD Anderson method, which is closer to what we do here, uses less, smaller margins. And they showed that the quality of life was much better and toxicity was much less with the smaller volumes. And in fact, that led to a survival difference. And it was thought that the survival difference was being driven by decreased toxicity from radiation, and the patterns of failure were the same. So the smaller margins did not lead to a different pattern of recurrent tumor. So I'm not going to get through all my slides, but that's okay. So this is a really, really neat study that was just published really recently, I believe, within the last few months or maybe a year, where we wanted to try to better understand this flare or peritumoral edema in GBM, because we know it can be a mix of edema or tumor. And so this used a radiomics approach. And there are low-grade gliomas, where the whole tumor is just flare, hyperintensity. And then we have cases of brain metastases, and we know brain metastases are very well circumscribed, and then any flare around them is just irritation from the tumor. It's not microscopic tumor in the flare, it's just purely edema. So what Archaya and colleagues did was they created radiomic signatures for the flare hyperintensity from low-grade glioma and brain metastases. And then they used those two signatures and applied them to the flare around a GBM. And you can see in this case here that this area posteriorly looks more like low-grade glioma, and the area anteriorly looks more like pure edema. And then they looked at patterns of failure, and lo and behold, the areas that looked more like low-grade glioma or microscopic tumor were the areas that were more likely to have recurrent tumor. So this is something we'd like to incorporate so we can more intelligently decide where to treat. There's another study, again, that I was involved with, led by Anne Martel and Andrei Muraviev, who was one of Anne's master's students. Again, looking at brain metastases and looking at some radiomic signatures, because we know about 5–10% of brain metastases, even small ones, will recur. Our control rate is 90–95%. That still means 5–10% will recur, and we don't know why. Some of them just are resistant. And so this looked at some radiomic features around the tumor and in the tumor that helped us determine which of these small brain metastases are more likely to fail. And then, we have to think of ways to try to get around that if we know it ahead of time. Maybe more likely to do surgery, or maybe try a higher dose. This is another study from the Netherlands. Again, it's a very difficult study that they did to try to look at all sorts of imaging, MRI, as well as PET imaging, with a stereotactic biopsy, where they go through the tumor and out the tumor, and they take biopsies along the way, and they can correlate that to exactly where they took biopsies on these various imaging sequences to see what are the best sequences for understanding what's tumor and what is just edema. So you can see they're out of the tumor here, but here they're in the tumor. And at the end of the day, the PET and some diffusion signals best correlated with actual histology. And this is an MRI, but this is just came out within the last couple of weeks. Again, it's using PET to try to determine what's the most metabolically active area of a GBM. Because if you ask radiation oncologists, why aren't you curing more patients, or why aren't you controlling things for longer? A lot of us have the knee-jerk reaction of, well, we didn't use enough dose. We didn't have a high enough dose. And people have tried to go beyond 60 gray in GBM to 70, 75, 80 gray, and it hasn't made a difference, probably because we're not intelligently targeting the areas that are the most resistant. And so this used PET imaging to define the most metabolically active area. They escalated to 75 gray, and they showed it was relatively safe, but it's not a randomized trial that can really compare to non-dose escalated. But we're trying to get there with the MR-Linac to use MRI to determine the areas that we could potentially boost to see if that in any way helps to delay the recurrence of the tumor. And the group in Michigan has really been the group that's probably developed this most. this is a high B-value DWI. This is a B3000 diffusion weighted image. And we know that typical diffusion data from 0 to 1,000 is really confounded. The areas of edema with or without microscopic tumor, the diffusion isn't very good at separating out different areas. And so what they have shown in some previous studies, a group in Michigan, is that if you use a B equals 3,000, that really highlights the true diffusion restricted areas that are more likely to be active tumor, and they're using that to define their boost volumes. And you can see that the red here, or the enhancing tumor, is in the middle here, but these high B value enhancing areas are outside of the visible tumor, but are clearly still active tumor. And so you would want to make sure you include that in your radiation volumes. Okay, that's some time. So I'm going to get into looking at different aspects of diffusion and then CEST. A lot of the diffusion stuff, this is all from Colleen Bailey, who's really leading this effort. And I'm trying to help from a clinical standpoint in terms of how can we use the diffusion image data in prostate cancer specifically. And we have people working on it in brain as well. So as you guys know, if you do a 0 and 1,000, you can fit a straight line and you can calculate the ADC. But we know that that's not the best fit, that it's actually a bi-exponential model, and that some of the nonlinear areas in the low B values are from vascular perfusion in small blood vessels. And that's the IVIM model. If you acquire more low B values, you can model that properly, and you can actually get a perfusion fraction. So how much of the area is actually has microcirculation essentially. The mid B range values are ADC, and then we know that the high B values, as I said, is more driven by membrane restriction and can highlight specifically a tumor better than just the standard ADC model. And Colleen has worked a lot on this model called Verdict, which again tries to more intelligently model a diffusion experiment in MRI to better understand both perfusion and diffusion as well as cellularity. So this, again, you guys probably know more about the details of Verdict than me. This is a prostate. There's some tumor posteriorly here, and the Verdict modeling from a diffusion sequence can extract cellularity, permeability, and some vascular perfusion parameters. And so we're trying to see, can this more intelligently help identify prostate tumors and the response of prostate tumors to radiation. And the thing is, we're treating some prostate cancers on the MR-Linac, which is great because we have patients in an MRI every day for each of their fractions of radiation. So we're repeating all this diffusion data, and we're seeing how things change over time. And we've only done a very small number of patients, but Colleen has worked at identifying the area where the cancer is most prominent in this case, and seeing how do things change over time, and is there a pattern that we'll eventually be able to find that will predict who's going to be cured of their prostate cancer and whose prostate cancer might come back locally. Again, it's early days, but there's a lot of legwork in trying to get good enough imaging on the MR-Linac to try to understand this. So we're looking at changes pre-treatment to during treatment. The other cool thing that we're doing, which at the end of the day may end up meaning nothing, is we can actually do diffusion data before we turn the radiation beam on, while the radiation is actively being delivered. It takes about 10 minutes, and then right after the radiation is delivered. And there seems to be some subtle differences in some of the diffusion and perfusion parameters. Some of that may be all technical, but there is a chance that some of that is truly biologic. And if we can understand that, that would be, really a neat thing to try to understand. Just gonna take a drink and pause for a sec. So this is just another utility of IVIM perfusion. Again, this is from a pretty basic diffusion experiment. This is not to directly guide radiotherapy, but we commonly treat brain metastases with radiosurgery, high doses of radiation. And about 5% of the time, we can cause something called radiation necrosis, where the tumor is gone and ablated, but there's a late 3 month to 3 year inflammation with necrotic tissue developing in that area. And there's another 10% of patients where, despite the radiosurgery, the tumor grows back. But when we see an area here that looks bigger on a T1, a contrast-enhanced T1 scan, we can't tell the difference between tumor that's growing back and necrosis. They look identical, and there's no good way to know. You can go in and cut these out, but we don't want to put everyone through surgery, and some places in the brain are surgically just not accessible. So I did this study a few years ago where we looked at perfusion, IVIM perfusion, which is, again, a pretty simple model to be able to do. And we showed that when there was higher perfusion in the small blood vessels, which we expect with active tumor, that predicted for tumor, versus when it was homogeneously low perfusion, that was indicative of necrosis. And this was a study done in about 11 patients where we did the perfusion and then they actually got their tumors resected. So we had pathology confirming exactly what was going on in this area, whether it was truly necrosis or recurrence. And we did show that IVIM was useful, and actually ADC did not separate the two in any way. And we're using a lot of other diffusion-weighted IVIM parameters of the enhancing tumor, of the peritumoral edema. This is a GBM, led by one of our neuroradiologists, Pejman Marilani. And we're trying to come up with diffusion and perfusion signals and how those change from baseline to day 10 of radiation to day 20, and see if that can predict who's going to be controlled for longer and who's not. And that has some prognostic value, and hopefully, we can use it to actually affect how we treat, so that patients where patients that have a signature, a perfusion signature that predicts for early recurrence, we can maybe change our strategy. But again, it's early days in trying to understand what these signatures are. And that brings me to the last sort of section, which I'm glad, I think I'm close to being on time. And that is of CEST MRI. The people I think on this, some of the people on this call, Rachel and Angus and Greg, are the experts in CEST. I have tried to understand it from a technical standpoint. Again, my PhD is in cardiac MRI, so I do have a physics background. I somehow still have a copy of Kim Desmond's PhD thesis, and about every 6 months, when I have a little bit of trouble sleeping, I decide that I want to really understand and learn CEST MRI and how it's done and what it means. And I read through it and I think I have it, and then a few weeks later, I don't have it. So we like to, clinically, it has been very helpful, and there's a big program with CEST MRI. I'm not going to be the one to explain some of the basics of it, but essentially it's looking at certain proteins, amide proteins particularly, by exciting at different resonance frequencies, having that amide hydrogen exchange with water, and then you can saturate it out, and then you can detect how much of this protein is there. And these proteins are more present, these amide proteins are more present with active tumor and with tumor rather than non-cancerous regions. It's a real basic sort of explanation of my understanding of CEST MRI. And going back, it's, been about 6-7 years at least now that really CEST has shown to have a lot of value in the brain in particular. And so one thing that we got up and running is, again, this idea of radiation necrosis versus tumor recurrence, and we can't tell the difference. And even though I showed with IVIM, there is some separation, we found that CEST is actually much better at separating areas of tumor recurrence versus areas of pure radiation necrosis. And as Rachel is very well aware, because we email all the time about this, once a week, once every other week, we have a case where we don't know what to do. Do we need more radiation because it's recurrent tumor, or do we need to use steroids and Avastin because it's pure radiation necrosis? And we don't know. And so they showed that You can use CEST to reliably separate these two, and this is making a big clinical difference in how we manage patients. There's also CEST and MT parameters within tumors of glioblastoma that are associated with tumor progression. So we're finding signatures to help predict who's going to progress earlier versus later. This is from Angus looking at different CEST signatures of different types of brain tumors. Again, showing that the more active and higher perfusion, and can definitely correlate, and we can see that on the CEST. Rachel and Angus have really put in a lot of work, because a lot of the work on CEST was at 3T or diagnostic scanners, and we now want to be able to do this on our MR-Linac, because we're having patients being treated every single day, and we're able to acquire whatever we want. But the MR-Linac, the MR portion of the MR-Linac is not like a diagnostic scanner. The gradients are not as good, and the fact that there's a Linac surrounding it makes the performance just not as good. And so Rachel has and the team have put in a lot of work to try to get all the advanced imaging, DWI, perfusion, and CEST up and working on this MR-Linac. And this was recently published, and we can do it. It's not quite as robust, but it can definitely be done. And now that we've shown it can be done reliably, we can start to use it and test it and see how it can affect our management of these patients. Again, just studies looking over time of CEST signal changes within the tumor and outside the tumor, and we are seeing changes and differences, and we have to start understanding exactly what it means, and then figure out a way to incorporate that into our management plans. And I think this is my last slide, or one of my last slides. At the end of the day, whether it's prostate or whether it's brain, the idea is we have our standard imaging, anatomical imaging. We have tumor, we have some flare around it, which may or may not be tumor. We want to pull in diffusion information, relaxometry, perfusion information. There is some people working on bold imaging, which I didn't talk about. And we want to use that to really define and personalize our treatment for each patient. Is there an area that's resistant that we need to boost higher than 60 gray? What areas outside of the gross tumor do we need to include, or do we not need to include? And how can we all do that all on the MR-Linac, for example, so that we can adjust daily and personalize treatment and minimize toxicity while trying to maximize tumor control. So that is all. That is all I had.
Fantastic. Thanks very much for the talk, Jay. I think we can open the floor to questions. So feel free to put a question into the chat box or use the raise hand feature. Maybe I'll start us off. Oh, I see we have a question from Wilfred. Yeah, please go ahead and unmute yourself.
Hi, Wilfred.
Oh, hi, Jay. How are you doing?